when we're talking about molecules, I say different names. It doesn't mean that that name belongs to just a specific molecule. For example, we talked about cadherins, and then we saw that different types of cadherins. Just like that, integrins, there are several different types of integrants, and they have different properties. So let's look at the properties of different integrin molecules. Here, for example, we have said that integrin is made up of two units, alpha and beta units, and these are dimers basically. So let's look at some of the combinations of alpha and beta subunits when they come together and they form an integrin molecule. What are some of the properties of that molecule? So alpha 5, for example, when it pairs with beta 1, it can bind fibronectin. Here again, please notice that I have used the word ligand because we are going to consider integrin as a signaling receptor because it shares many features with our traditional signaling receptors. So it is going to, the ligand for this particular combination is fibronectin, and it is ubiquitous, meaning that it is found in almost all cells. Alpha 6 beta 1 combination, it binds laminin. Now kindly notice that the beta subunit is the same. It's the beta 1. Just changing the alpha subunit from 5 to 6. Now the binding properties of integrin dimer has changed. It is, although it is ubiquitous, it now will bind laminin rather than fibronectin. Alpha 7 beta 1, it binds laminin, but it is specifically found in muscle cells. Alpha L and beta 2, it binds Ig superfamily. We have talked about Ig adhesion molecules which have immunoglobulin-like domains. This type of integrin molecule is found on white blood cells. We will talk about that later, but just please keep in mind that it is beta 2 and alpha L subunits. Alpha 2 of these units come together and form alpha L and beta 2 integrin molecule. Alpha 2 and beta 3, they bind fibrinogen. It is found on platelets. Alpha 6 and beta 4, they bind again laminin, and this type of integrin molecule is present in hemidesmosomes. We have seen that we have already seen structures of hemidesmosome. The cytoplasmic domain of this combination, beta 4, will bind, as we know, intermediate filaments and not actin filaments. Other beta subunits generally bind, pretty much all of them bind actin filaments and not the intermediate filaments. This guy is the exception. ECM is recognized by multiple integrins. At least 8 integrins bind fibronectin, different combinations of them, and at least 5 bind laminin. A variety of human integrin heterodimers are formed from 9 types of beta subunits and 24 types of alpha subunits. So there are many different combinations are possible. This diversity is further increased by alternative splicing of some of the integrin RNAs. So although there's 9 genes for beta subunits and 24 genes for alpha subunit, but we can not only there's a combination diversity, any of beta can combine with any of alpha theoretically. Additionally, the diversity is generated by alternatively splicing the messenger RNA for coding for integrin molecule. Beta 1 subunits are found on all vertebrate cells. They form dimers with at least 12 distinct alpha subunits. Mutant mice that cannot make beta 1 integrins die at implantation because you can see it is a very important combination. It is present in all cells, and it is playing a very important role there. Whereas mice that only are unable to make alpha 7 subunit, this is the alpha 7 subunit, these mice do survive, but they develop muscular dystrophy. And also, if rather than deleting alpha 7 subunit gene, if we delete the laminin gene, it still produces the similar phenotypes. It also this data suggests that this particular integrin is most likely binding this laminin. The beta-2 subunits form dimers with at least 4 types of alpha subunits. They are expressed exclusively on surface of white blood cells. They have an essential role in enabling these cells to fight infection. I'll show that how this system works. The beta-2 integrins mainly mediate cell-cell rather than cell matrix interactions, binding to specific ligands on other cells such as endothelial cells. We have seen a movie also that how white blood cell is rolling because of weaker interactions, and ultimately I had said that that white blood cell has to bind the endothelial cells very firmly in order to cross the endothelial cell barrier and go to the site of infection. Beta-2 integrins enable white blood cells to bind endothelial cells at the site of infection and migrate out of the bloodstream. In humans with genetic disease called leukocyte adhesion deficiency, abbreviated LAD, unable to synthesize beta-2 subunits, their white blood cells suffer repeated bacterial infection. I meant that humans with genetic disease called leukocyte adhesion deficiency are unable to synthesize beta-2 subunits. Consequently, their white blood cells lack the ability to stick to site of infection, and these people suffer repeated bacterial infections. The beta-3 integrins are found on a variety of cells, including platelets. These integrins bind several matrix proteins, including fibrinogen. Platelets, when they interact with fibrinogen during blood clotting, they basically form these plugs to stop the bleeding. In a human disease called Glanzmann disease, these people have basically a deficiency in beta-3 integrin, and they bleed extensively. So let me show you with the help of diagram, how the system is working. So as we have said that initially when there is an infection or there is a wounding, the endothelial cells, they first of all start producing or start putting a specific selectin on their surface. Selectins, remember, mediate weak interactions. These selectins bind a specific carbohydrate on the surface of the white blood cell. And now they are going to slow the white blood cell. White blood cell is moving in the bloodstream, and the white blood cell is going to start rolling rather than rushing through. Ultimately, what happens is this interaction, binding of selectin to this particular selectin ligand, activates a specific type of integrin present specifically on the surface of white blood cells. As we talked about, it is the alpha 7 beta 1 integrin. Once this is activated, as we will see in the next slide, once this integrin is activated, it binds another specific molecule on the surface of endothelial cells. So this is our adhesion molecule, which is going to bind another molecule present on the surface of endothelial cells. These molecules are also again expressed at the site of infection or where there has been a damage. Now this interaction has activated this integrin. In previous slide, you saw that it was pinkish color, meaning this particular integrin was in active form. Now in active form, this integrin will bind its substrate or its ligand very tightly. This is a weak interaction. This is a very strong interaction. This will result in basically sticking off this particular cell at a particular site. When this white blood cell is firmly stuck on the surface of endothelial cells, it ultimately basically starts to move through the intracellular space and goes to the site of infection. So we have seen that integrins play a very important role in attaching cells to the extracellular matrix. We have also seen that integrins also play a very important role in cell-cell adhesion in case of white blood cells.